Welcome to today's lecture on marsupial stress and disease. The objective of this lecture is to understand that stress and disease represent major conservation challenges for Australian marsupials. Today we will discuss the stresses that marsupials face and cover case study examples of marsupial disease. From information provided in this lecture and from your own reading, you should gain an understanding of the ultimate and proximate major stresses that marsupials face. You should acquire knowledge of what is quantified to assess stress responses and stress levels and how this is quantified. So the methodology used to quantify stress. And you should appreciate that stresses and responses to stress can have flow-on effects for both individuals and populations. And you should be able to describe some examples. So why are stress and disease important to understand and study? Well, many Australian marsupials are threatened species, so more than 40% are of some level of conservation concern. We recognise that in order to manage both in situ and ex situ populations effectively, it is important to understand how marsupials are responding to threats and how these threats may induce stress upon the animals. Stress physiology, which is the study of the response of animals to challenging stimuli, is a key approach in conservation physiology and can be used to characterise the physiological responses of wildlife to threats. The need for effective wildlife health investigations in this fast-changing world with growing concerns about biodiversity loss means that we need to understand how disease is going to threaten fragile populations. So the detection of emerging wildlife infectious diseases is also critical to recognise and manage in relation to the adverse impacts that these diseases can actually have on human health. On marsupials specifically, or rather generally, there is an extensive body of literature that highlights the complex roles of the stress endocrine system in the physiological responses of wildlife to stresses. However, in comparison to other taxa, in marsupials specifically, less is known about these responses, especially in relation to conservation-related stresses, and therefore we lack a bit of knowledge about the conservation implications of the marsupial stress response. This is concerning because marsupials are exposed to a suite of potential stress factors such as habitat loss and increased predation pressure from introduced predatory species. Marsupials are also unique. They occupy a very niche, a very specific evolutionary niche. So investigation of how marsupials respond to stresses can provide insights into the evolution of the physiological stress response more generally for animals and across different taxa. And this sort of area of research is an ongoing area in evolutionary ecology. So Australian marsupials possess many unique physiological characteristics, such as adaptations to specific climatic envelopes. And from that, this means that, that there may be facilitation of distinct physiological stress responses within this group of mammals. So stress and the stress response. Stress can either be simulatory, so this is optimizing physiological symptoms for action. It can be preparative, priming systems for action, or it can be inhibitory. So this is especially what we see in chronic stress, and this can cause permanent dysfunction of the endocrine stress response and lead to the suppression of critical functions such as reproduction. The stress response is a complex and vital physiological mechanism that enables individuals to cope with stressful situations. So it is an adaptive response. It involves the production of neuroendocrine mediators such as adrenaline and glucocorticoids, and it is essential to maintain allostasis through exposure to a stressor. So the hypothalamic pituitary adrenal axis, or the HPA axis, is the neuroendocrine pathway underpinning this physiological stress response. It's recognised to be highly conserved across vertebrates, including eutherian mammals, marsupials, and monotremes. The HPA axis operates as a negative feedback system. So secretion of corticotrophin releasing hormone from the hypothalamus will stimulate release of adrenocorticotrophic hormone, or ACTH, from the pituitary gland. ACTH then promotes the release of many neuroendocrine mediators, including glucocorticoids, from the adrenal axis. So glucocorticoids will be the focus of today's lecture when we talk about stress and stress response. Glucocorticoids, which mainly, when we talk about them, we're mainly either referring to cortisol or corticosterone, have complex interactions with virtually all other biological processes. Glucocorticoids are acknowledged as practical endocrine indicators of stress and are being integrated into wildlife conservation worldwide. So like eutherian mammals, glucocorticoids in marsupials are known to regulate a suite of biological processes from protein, carbohydrate, and fat metabolism to essential renal, neurological, cardiorespiratory, and reproductive functions. There are several unique characteristics which may constitute evolutionary adaptations to stresses that marsupials face in many of the harsh environments that they occupy. So as an example, in some macropods, it seems that the influence of glucocorticoids on renal function is important in the control of the salt and water balance. In relation to glucocorticoid quantification in measuring stress responses, endocrinology has undergone a revolution since the first study on glucocorticoids was performed on an Australian marsupial in 1964. So this initial study involved looking at glucocorticoid levels in the blood of brush-tailed possums. Invasive terminal sampling collection was commonplace in the 1960s and 70s, but fortunately, these methods have been superseded by non-invasive techniques because they're obviously not always practical or ethical, especially when we're studying cryptic and endangered species. So over the last 30 years, minimally invasive techniques have been used to measure glucocorticoids and their metabolites. And these methods have been broadly applied to identify stresses to wildlife. So assays have been validated for use in different wildlife species to allow glucocorticoids to be quantified in still blood. So in some instances, it's still necessary to measure blood, but this can be achieved without having to sacrifice the animals and with only a small amount of blood. But more commonly, we tend to adopt invasive, completely invasive methodologies that will involve glucocorticoid analysis of feces, urine, or hair. So over here you can see a pie chart of the different stress parameters and methods used in marsupials. And it's evident that most commonly measured stress parameter in marsupials has been through the use of fecal glucocorticoid metabolite analysis, so using enzyme immunoassays. So when I refer to fecal steroid analysis during the lecture, this is what I'm referring to. Excuse me. Key factors to consider when integrating stress physiology into marsupial conservation include considering what is to be measured and ensuring that the protocols are actually standardised and validated. So circulating glucocorticoids in blood should be collected within 2 to 3 minutes of capture. But we recognise that immediate collection is rarely possible in marsupial field studies. So tracking and handling and sparsely distributed and often nocturnal animals with minimal resources in challenging terrain means that it's really not often that you're going to have the opportunity to collect blood immediately upon capture. Because of these challenges, measuring steroids in the faeces of marsupials has proven to be a very popular method. So the relative value of fecal analysis on fresh versus old samples by quantifying decay rates showed that glucocorticoid metabolites were relatively stable in bilby faeces for up to 19 days. So that's encouraging and means that this methodology is quite suitable to this species because it eliminates that need to be sampling immediately. But obviously validation needs to be done for each species owing to these differences in specificity in decay rates. So yes, non-invasive methods, they need to be validated for each species. As metabolism and excretion of steroids will differ significantly between species as well as sexes. So this validation is typically carried out using an ACTH challenge, which involves inducing a physiological change in circulating glucocorticoid levels via injection of ACTH. And then there is a process of an evaluation of how these changes are reflected in the faeces. So a study has applied this approach to validate the use of fecal cortisol analysis to assess the activity of the HPA axis in koala feces. And it was determined in this study that, in fact, it was a suitable method for assessing the adrenocortical status of koalas in wild populations. So essentially that you could use this methodology to measure stress levels in koalas in the wild. So because glucocorticoids play a vital role in regulating metabolism, activity, reproduction, and response to environmental challenges, there's obviously going to be natural fluctuations in glucocorticoid levels. And it's been recognised that they will vary due to diurnal rhythms, reproductive status, and season. So this degree of naturally occurring variation needs to be considered in order to avoid misinterpretation of normal fluctuations as a significant physiological response to a stressor. It's important that study design and analyses are acknowledging and trying to count for these this natural variation, and it's obvious that to consider this is essential when we think about it in relation to incorporating stress physiology into marsupial conservation. So the stresses for marsupials, there is a range of broad categories of stress factors that have been identified, and these include habitat loss and fragmentation, captivity, translocation, and climate. So each of these ultimate stress factors, which are essentially the root causes, may entail a number of potential proximate factors. So the more immediate factors that are influencing animals. Wildlife are exposed to biological and anthropogenic stresses, both of which can have very important implications for conservation efforts. So today we'll focus on anthropogenic stresses and disease, which is recognised to be a biological stressor that may be exacerbated by human activities. So as an example of biological ecological stresses that naturally occur for marsupials, this includes reproduction, social grouping, disease and illness. And in terms of anthropogenic stresses, we're talking about things like climate change, habitat alteration, capture and handling, and translocation. So both biological and human-induced stresses are obviously going to have implications for both ex situ and in situ conservation. So talking now about some of these stresses, habitat loss and fragmentation may act as stresses through proximate mechanisms such as reduced resource availability, increased competition, altered behaviour and social arrangements, and disease stresses associated with changes in population density. It's recognised that marsupials tend to experience greater physiological stress at higher densities compared with lower densities, as has been measured by plasma or blood glucocorticoid levels. Habitat fragmentation can increase stress through the creation of more habitat edges. So in squirrel gliders, it's been found that individuals living in edge habitats actually have higher cortisol levels compared with conspecifics that live in more interior habitats. So this might have something to do with proximity to humans and human activities. Differences in physiological stress between marsupials in fragmented compared with continuous habitat may also underlie differences in immunocompetence and infection patterns. So flow-on effects of that stress response influencing how individuals cope with disease and illness. Captivity is a stressor, specifically in relation to ex situ conservation programs, those being ones that rely on captive breeding to supplement wild populations. Captivity can alleviate potential stresses that individuals might face in the wild, such as resource limitations, exposure to climatic extremes, and exposure to disease and predators. The captive environment is also going to entail a range of biotic and abiotic stresses that would not be faced in their native habitat. So the factors include frequent human contact, abnormal social grouping, confinement, inability to exhibit natural behaviours, artificial lighting, as well as different smells and different substrates that they're living on. So using fecal steroid analysis, the response of captive numbats, the stress response of captive numbats basically was assessed in relation to 20 key stresses, and these were classified by different categories, including conspecific interactions. So this is in relation to the introduction or separation of familiar and unfamiliar animals. There was an environmental category, another category that involved handling and health. So for example, any injuries or procedures that were performed on the animals. And then another category included anthropogenic disturbances, so such as zoo events, because this captive breeding colony is held at the zoo. So zoo events or maintenance activities near or within the enclosures. It was discovered that handling, conspecific interactions and health-related factors were all found to elicit a significant physiological stress response. This absence of response to anthropogenic disturbances was concluded to be due to the fact that these individuals were habituated to to humans and human interaction. But something that wasn't covered in this study was how these stresses compare to what would individuals would incur in their natural environment. So it wasn't really any assessment of whether captive numbats were more stressed than wild numbats generally. In sugar gliders, so these guys are social animals, and captivity can alter or provide abnormal social groupings, which may lead to social stress. There was an assessment of stress responses in sugar gliders to other individuals or their scents, which can provide valuable information for the management of social groups in captivity. For example, how sugar gliders should be housed in captivity. So this study on sugar gliders investigated social status and stress in male sugar gliders, captive male sugar gliders. Basically, the odour of a dominant male from another group was found to elicit an increase in plasma glucocorticoids. So, though there was no changes in stress parameters of male captive sugar gliders when they were exposed to odour from a castrated male or a female. So it seemed that the scent of a socially dominant male sugar glider from an unfamiliar colony did elicit a stress response, but this was only for dominant males and not for castrated males or for females, which isn't that surprising because sugar gliders sugar glider groups tend to have one dominant male. So yes, but again, not any investigation as to how that is reflected in the wild and how dominant males of wild social groups might respond to the sense of other dominant males. The flip side of variation in social groupings is that social marsupials may be stressed by isolation induced by captivity. And a study on tamer wallabies challenged individuals with changed social conditions. So they moved from a grouped housing situation to isolation. The faeces was collected in 24-hour intervals during this study period and analysed for glucocorticoid concentration. And then it was found that there was a significant increase in corticosteroids concentration upon isolation. So these levels remained above initial levels throughout the study period, suggesting that fecal corticosteroid concentrations may in fact be a useful indicator of a change in animal well-being as it is sustained over a period of time. Wildlife in captivity do provide surrogate study specimens due to the logistical difficulties of working with free-ranging populations. So this is especially true for many marsupials, for example, the shy and elusive numbat. So wildlife and wild marsupials held in captivity may provide a greater understanding of how these animals respond to captivity. And in so doing, we can sort of interpret experiments done on captive animals or investigations done on captive animals in a more enlightened way. This, as well as measuring and monitoring stress in captive marsupials, will help to improve conservation outcomes. And this is, obviously this is especially for those marsupial species that rely on captive insurance populations. So capture and handling are relatively well-characterised stresses in eutherian mammals, particularly laboratory animals and livestock. A small number of studies have suggested that handling elicits a stress response in captive and wild-caught marsupials. So physiological indices, including blood levels of glucocorticoids, as well as fecal steroid levels, indicate that there is an acute stress response to handling in some species, including koalas, numbats, and wombats. It's also been discovered that behavioural reactions to capture and handling are not always accurate indicators of physiological stress. So it's been shown that reactions of captive wombats will wane with regular handling, but sort of an increase in sustained fecal steroid levels indicate that their physiological stress response actually doesn't diminish. So this impact of capture and handling stress in marsupial conservation obviously needs to be addressed, and we need to think about ways in which this stress can be managed and mitigated as much as possible. Climate is another stressor for marsupials. So there is little research into the stress response of marsupials to climatic extremes, and it seems to be assumed that marsupials possess adaptations to deal with extremes, because a lot of species are adapted to harsh conditions. I think this lack of attention is a bit concerning, because stresses related to climate change are a significant threat to biodiversity, and often approached from the standpoint of how climate change alters the ecology of the system. But there's sort of, a lack of attention about associations between climate change and wildlife stress physiology on individuals. So climate can affect marsupial stress and health. It's known that quokkas experience seasonal debility in relation to elevated cortisol levels, and they will be, at these times, they will have reduced body mass, they will be dehydrated, and their parasite infection loads can be higher. So, and then koala studies have found that Queensland individuals at the arid edge of their range also tend to have higher fecal steroid levels compared to individuals that are in more temperate areas. And this is especially the case during drought. So, and it's believed that so individuals in the arid edge of their range during drought times is obviously going to be experiencing a lot of stress due to variation in dietary composition, as sort of ongoing climate change issues result in more extreme climatic events and make climatic conditions more stressful for individuals. Climate change has been suggested to be a contributing factor to past, present, and will be future range contractions in marsupials, and has been responsible for local extinctions of Australian marsupials. So yes, I think that this area is a little bit neglected and probably currently needs a bit of attention, not only to see how climate affects stress levels, but the sort of ongoing implications of that in relation to populations and the sustainability of populations. So the physiological response of the critically endangered Barrow Island Euro to prolonged drought. So this was an 8-month study that looked at how lack of water and nutritional stress influenced plasma glucocorticoid levels. So basically, it was found that as there was increasing plasma glucocorticoid levels, there were hematological changes. So there was a decrease in eosinophils, which are disease-fighting white blood cells. This suggests that there was a downregulation of the part of the immune system that is responsible for fighting macro parasites during drought conditions. So immune function was essentially suppressed as a result of elevated corticosteroids. And I think this study is important in demonstrating that although marsupials possess many unique physiological adaptations, they are obviously not immune to the impact of climate stresses. So now just sort of exploring some consequences of stress. Acute versus chronic stress can have different effects on fitness, and it's important to understand the consequences of acute stress. So this is stress that we think of in relation to capture and handling versus chronic stress, which is more like confinement due to captivity. There may be an interaction between responses to acute and chronic stress. So in bilbies, individuals experience long-term stress associated with injuries or health issues, demonstrate a greater stress response to acute stresses. It's crucial that links between glucocorticoids, biological function and fitness are fully characterised to understand the impact of stresses on Australian marsupials, particularly those species that are of conservation concern. In this way, we need to understand what normal versus potentially detrimental variations in stress levels, what they are for different species. And so we need to also, in order to determine whether a stressor poses a significant threat to an animal, we need investigations into associations between, specifically between stress levels and biological functions, including immune defences, disease dynamics, reproduction, and fitness. So thinking about how stress can influence reproduction, this is important in marsupial conservation because chronic stress is known to limit breeding, and obviously this is then going to influence the sustainability of wild and captive populations. So chronic stress associated with breeding in marsupials can be associated with fatal pathology. So this is referred to as seminal parity, and we will discuss this again in the reproduction lectures. But basically what we see in some marsupials, such as the Antechinus, males have a negative feedback system that regulates glucocorticoid release. In males, the negative feedback system that regulates glucocorticoid release is actually abolished during the breeding season. So this tends to result in a massive male die-off. So basically all the males will breed, then die. And what's going on is that there is a decrease in plasma corticosteroid binding globin, or CBG, which results in an increase in biologically active cortisol to levels which are immunosuppressive. So males then, as determined by post-mortem, it's been shown that males suffer severe ulceration of the gastrointestinal tract. There's an increased invasiveness of microorganisms and parasites, and also liver pathology has been associated with listeria. So this decrease in CBG basically results in an immunosuppressive way, and then males succumb to disease and parasites and eventually die. It's been found that seminal parity can be affected by environmental factors. So captive and wild brush-tailed phascogales differ in their physiological stress response to reproduction. Captive males seem to escape the seminal parious fate of their wild counterparts. So what you can see here in these figures is cortisol levels of wild males during the breeding season, where there's a significant elevation, and this is the same period in captive captive males, where they almost tend to show a decrease in cortisol levels. So these insights indicate that conservation success for semmelparus marsupial species may rely on mitigation of proximate stresses associated with reproduction, such as nutrition and energy deficits. So when you remove those environmental effects of food availability under captive conditions, then it seems that die-off is not operational in this species. But even in those species that do not display seminal parity, stress can influence breeding success, and stress physiology may be highly informative for sort of addressing troubleshooting reproductive failure in breeding programs and just the sustainability of wild populations. In interpreting stress physiology in reproduction can be problematic given the complexity of the interactions between glucocorticoids and reproductive hormones, difference in protocols, methods and techniques that might have been used across species and even within species. There's obviously going to be species-specific differences when we think about captive populations. Length of time in captivity has to be taken into account, as well as different characteristics of the captive environment, as well as, of course, the nature, frequency, duration, and severity of stresses that individuals are subject to. But there is an extensive body of literature indicating that stress can have a significant impact on reproductive success. And understanding stress physiology when we're looking at the reproductive performance of marsupials is obviously going to be beneficial to both in situ and ex situ marsupial conservation. So it's been shown that female honey possums are actually vulnerable to chronic stress in captivity. And then this is the critical factor that will impede their reproductive success under those conditions. So obviously this is a serious issue for populations that are relying on captive breeding programs to supplement wild populations. Okay, so moving now on to disease. Disease is a stressor, but stress can also initiate disease. So marsupials cohabit many environments with humans, and therefore diseases common to marsupials are fairly well documented because the interest has lied in how these diseases might influence ourselves. Marsupials are subject to ectoparasites, which include ticks, lice, mites, fleas, and can be identified by areas of the skin that are crusting or might be red. And these tend to occur in the groin or leg pit regions, or sometimes it's recognised as orange crusts around the pouch. So there's a few examples there in wallabies, and the middle one is actually a echidna, but the other two are different species of wallaby. Mange, which is caused by the Sclerotes mite, has been recorded in numerous species, including koalas, wallabies, and bandicoots. However, common wombats are the most readily reported individuals or species affected by mange, and infestation with this parasite has resulted in significant population declines. So mange in wombats has been associated with overcrowding, habitat degradation, and high density of foxes or feral dogs. So some association between stress there and the frequency of mange. When more than 30% of the skin surface is covered by this mange crust, wombats have a marked reduction of muscle mass, and there is an atrophy of fat. So this is the nasty little mite here that causes mange. And here we have this is a common, this one here is a common wombat, and this is a southern hairy-nosed wombat with that are infected by mange. Marsupials are also subject to endoparasites. Endoparasites are extremely diverse, many are cryptic, so we don't understand their diversity completely, but it is getting better as there's different molecular ID methods developed. There's estimated to be more than 600 species known from marsupials and monotremes, with identifications mostly being performed on post-mortem and fecal examinations. Parasites of prey species can actually confuse things when investigating endoparasites of carnivorous marsupials, because passing prey through the gastrointestinal tract, it can't be clear whether that endoparasite was from the prey species or is one belonging to the carnivorous marsupial. Types of endoparasites include trematodes, which include the flatworms, nematodes or roundworms, cestodes, which are tapeworms, and protozoa. So these are single-celled parasites, such as toxoplasmosis. And toxoplasmosis is actually quite a common infection to marsupials. Marsupials tend to be highly susceptible to this parasite. So this parasite can actually infect any warm-blooded animal, including ourselves. Looking at a couple of case studies of marsupial disease, we'll start with koala chlamydia. So the general belief is that chlamydia has been a component of the koala's natural history. However, recent molecular investigations of the bacterium that causes chlamydia has suggested that some koala infections may stem from introduced domestic livestock. The spread of chlamydia in adult populations is through reproduction and social behaviour, whereas joeys tend to catch the disease from their mothers. So when the infection establishes in the eye, so chronic conjunctivitis can then lead to corneal scarring and eventual blindness. So that's displayed up the top here in different levels or different degrees. When the infection establishes in the urinary tract, so this can include the urethra, the bladder, the uterus and kidneys, there's a number of different issues that can arise, and this can cause severe pain for the animal, and it can actually result in excessive urination and or urinary incontinence, leading to wet bottom. So if we just flip back to the previous slide, here you have a koala bottom of a healthy individual, and this is the bottom of a koala that's infected with chlamydia, and is the rump is stained due to that incontinence caused by chlamydia. When the infection establishes in the reproductive tract, inflammation in both females and males can lead to scarring and infertility. Chlamydia can also lead to secondary skin infections and ulceration. And it's fairly nasty. It gets nastier, so it's not only limited to the classic body sites, but it has been reported that it can also lead to fatal pneumonia, polyarthritis, as well as colonisation of the gastrointestinal tract. So unfortunately, infection rate has increased over the years. For example, the infection rate of a New South Wales koala population in Gunnedah has risen dramatically from 10% in 2008 up to 85% of the population being infected in 2021. And situations like this are actually playing out among koala populations across Australia, and threatening those populations that are already vulnerable to bushfires and habitat loss due to deforestation. Captive populations are at risk as well, and this disease does become prevalent when individuals are stressed through management or other disease and illness. So it's a pretty horrible disease, and it's affecting a lot of koalas. Every female koala that is infected with chlamydia will become fertile within a year or two. So even if they do survive, they're not going to breed, which is going to have obviously a pretty dampening effect on populations. Treatment and prevention. So the koala gastrointestinal tract is full of these specialised bacteria that are used for digesting eucalyptus leaves. And these bacteria are especially sensitive to antibiotic-induced disruption, antibiotic-induced disruption to their microflora. So this disruption to the gut microbiome can be fatal, and so this makes antibiotic use a very delicate balancing act. The quest for a vaccine is underway, and modelling has predicted the impacts of a vaccine on declining koala populations in Queensland. So it's been shown that with other threats remaining constant, modelling has predicted that a vaccine with 75% protective efficacy, covering around 10% of the koala population each year, and targeting young female koalas could actually reverse current population declines in 5 to 6 years. So evidence that the vaccine can have a positive impact on koalas seems to be mounting with each new study that is being published. So that's good news if we can get this vaccine underway and treating populations. Moving on now to the second case study, which is the Tasmanian devil facial tumour disease. So the wild Tasmanian devil population of Tasmania has been reduced by 77% over the last 20-30 years, and this is primarily due to the emergence of transmissible cancers, which is the devil facial tumours. So DFTD, or the devil facial tumour disease, is a rapidly fatal disease that culminates in these large tumours primarily on the face and mouth, which frequently metastasise to internal organs. So you might have noticed the cover image of today's lecture is one of those facial tumours on a Tassie devil. So it's believed that DFT1 probably originated from a cancerous Schwann cell in a female, whereas DFT2 arose of a similar cellular origin in a male devil. The rate of spread of the disease is variable as different barriers affect devil movements, such as rivers, mountains, dense bush, but it's been calculated to vary between 7 and 52 kilometres per year. So this figure here just shows the distribution of DFT1 and DFT2. So DFT1 is pretty much across most of Tasmania in the devil populations. So a remarkable feature of the devil facial tumour disease is that the cancer cell itself is transmitted between unrelated devils as an infectious pathogen, essentially a contagious cancer. So these malignant cells are transferred between individuals as a natural allograft, forming new clonal tumours. DFTD is almost 100% fatal, usually within 9 to 12 months of the presentation of a tumour, the individual will die. And weirdly, unlike most infectious diseases, DFTD typically affects the fittest individuals within a population. So by fittest, we mean those with the highest reproductive output, which is kind of counter. So you would expect that diseases are going to affect the older individuals more than younger and fitter individuals. So as DFTD is spread by injurious contact when individuals bite each other, transmission is the greatest amongst adults of breeding age, as injuries from intraspecific bites peak during the mating system. So this is why it's affecting sort of the fittest and most desirable individuals first. So mature breeding individuals of 2 to 4 years of age are usually those that are disappearing first from a population. A flow-on consequence of the reduction in local population density following DFTD is that growth rates of subadults actually increase. So this is because there is greater prey availability and nutrition to these individuals, which essentially allows a greater proportion of younger females to attain a sufficient body mass to breed in their subadult year, so in their first year. It's recognised that precocial breeding provides reproductive compensation to counter severe disease-caused mortality rates, but it is not sufficient to significantly slow the decline in population growth rates. But precocial breeding may result in rapid evolution of traits leading to a species that is more resilient to DFTD. So that would be cool if that happened. Safeguarding and prevention against DFTD. Removal of DFTD from wild population has been trialled essentially via a disease suppression program carried out in southeast Tasmania. So animals with signs of the disease were euthanized to determine whether this removal conferred any advantage to the remaining population. When this managed site or population was compared with an unmanaged site, analyses showed that this method was not effective. So quite laborious, but didn't have any sort of long-lasting effects on controlling the disease within a population. Other methods for securing healthy populations has involved translocations of healthy devils to islands and isolating populations behind barriers. But again, fairly laborious and sort of smaller scale type control or removal. Currently, there's no effective interventions for reducing or controlling the impact of DFTDs on a broad scale. An oral vaccine has been proposed as a safe and effective method for eliminating these diseases, you know, as sort of like an attempt to ensure the long-term survival of this endangered species. But it's not actually being applied yet. So just to finish up today, we mentioned that disease is recognised to be a stress, but how does disease and stress, how are they associated? Stress is implicated as a potential trigger for clinical disease in captive koalas infected by chlamydia. So we mentioned that previously. More generally, stress has been suggested to be a factor exacerbating toxoplasmosis infections, which is quite detrimental to Australian native fauna, including marsupials. In addition, hemoparasitic infections may be exacerbated by stress. This is known to play a role in the decline of the critically endangered woylie. But really, there's only been a small number of marsupial studies that have actually investigated this association between stress and disease. And we're still yet to investigate and characterise fully the health and conservation implications of physiological stress in marsupials. So another area of research that needs continuing investigation. So here is a list of some further reading. Some are provided on LMS and some are available in the library. And that's where I'm going to leave it. Thanks for tuning in.